We're back. How does it work? Yeah, bunch of question marks. What good's that going to do you? You get your samples, you figure out what methods you're going to use, usually involving mass spec, but there are other ways you can do this. And so there will be a lot of crossover with the methods that a good chemistry lab would use. There will be some crossover. There may be some things that you can detect better with an antibody or more sensitively with an antibody than you would be able to detect them by mass spec alone, for example. So yeah, lots of ways to do it. Then you're going to collect your data and then you're going to try and figure out, is this difference meaningful? And whenever you see the word significance in a science, if people are being careful, and that's kind of your job as a scientist to be careful, it should mean a statistical test is behind it. And there are a lot of different statistical tests that can be used, but basically, if you're talking about metabolites or proteins and you are not using statistical tests to eliminate the stuff that could occur by chance or that you can't say, 100% hand on heart is anomalous, then yeah, essentially these are garbage results. So it's stats that make the difference between sense and nonsense. Yeah. Okay, so you'll come up with your plan, you'll sample whatever it is you want to sample, a particular tissue or something. Next, you're going to try to separate. So, for example, I've done some work on lipidomics. And for that, you would take the cell, throw away all the cytoplasm and soluble stuff, and just purify the lipids. And there are some people that will purify the lipids from like the nuclear membrane, or they'll just purify Golgi's and then take all the lipids from that, or purify ER. There are ways to kind of do this. And you can actually see the composition of different lipids. You can even separate a bilayer. So you can look at the lipids that are on the outside versus the ones that are on the inside of a lipid bilayer, like a membrane. Yeah, so kind of neat. Then you're going to run them through the detector, which is usually going to be mass spec. And then here's our little mass spec data. There they are. And based on mass spec of standards, so if you're doing lipidomics, you would run various lipids of known, structure on there as standards, and then you would compare your profile to what you would see from these other ones. At the separation phase, you can also separate, for example, charged lipids, kind of polar lipids, non-polar lipids. So they're going to be lipids of different molecular mass, like sphingolipids can be fairly large. Things with different sort of head groups on them. Yeah, and you can sort those out and then analyze them further with the mass spec. Because mass spec is going to give you a mass for whatever you throw at the wall. But if you throw a bunch of things at the wall of similar mass, you kind of get a mess. So what you want to do is separate them out by charge or by size or something like that. I like to think of it as two-dimensional separation. So you're separating first on charge and then on mass. And so each of your little proteins is like one little data point, somewhere, some combination of charge and mass, or, electrophoretic mobility and mass. And that helps you narrow down exactly what it is you're looking at. Okay, so you can do some NMR for that, where you basically jiggle the atoms with a magnetic field in ways that makes them resonate. And you'll get little energy signatures that come off of that, and that can help tell you what atoms you're dealing with. Mass spec is going to be very widely used. And then it's going to be some type of multivariate statistical analysis that you use as the final thing, because you're going to have different things and you're trying to look often for a difference in profile. So usually with an omics type experiment, like metabolomics or proteomics or anything like that, transcriptomics, you don't just want to know is gene 1 turned up or turned down, because gene 1 is going to be connected to 50 other genes that make 60 other enzymes that make 200 metabolites between them, and there's going to be some subtlety there. When gene 1 goes up, it'll turn other things up or down or not move them at all, but maybe activate some things, deactivate some things like that. One way that we can look at this, and I think it's the most powerful one on here, is called principal component analysis. So this is a statistical way of taking a profile. Like, imagine you've got like a little bar graph. Like this one goes up, this one's a little bit up, and so you've got like a bunch of different markers and you've got different amounts of them. And so this is the cancer cell, and then this is the non-cancer cell. And you say, okay, that versus that, what's the difference? And so the difference is just going to be in these parts. It's basically a way of finding the things that vary together in a data set. And so you call these principal components, like things that co-vary within your data. It's an analysis that is highly mathematical, like in concept, but ends up being really useful because you can basically take a profile of an entire, metabolome and say, all right, it changes, and everything changes a little bit, but it changes in a characteristic way when I do this treatment, when I make it too cold, too hot, too acidic, too salty, whatever. And so you can get information on an entire program of different genes, maybe several different programs of genes that are going to be turned on, off, up, down, or not. And so, that's a way you can tease this out, is with principal component analysis. Okay. So targeted metabolomics is where you go in looking for a particular thing. Untargeted is where you're aiming for sort of global, like, let me know what's going on in this sample, not coming in with any preconceived notions. If you know you're putting in a drug that is supposed to like inhibit serotonin reuptake, something like that, and one of these SSRIs, then you can specifically go in and look for, okay, what are serotonin levels, in the cerebrospinal fluid after we put this SSRI in. Yeah, something like that. It's good. Or you can look and say, all right, I put my drug in, and I know that it can get broken down into this, and this other intermediate things. And you can say, all right, how much do I have of each one of these things? Because that's what you care about. You're trying to figure out, is this drug going to last long enough and not turn into anything dangerous or stupid as it gets metabolized? It's going to cause problems. And so that would be examples of a targeted approach when that might be better. Okay, LC-MS, you remember that? The LC is liquid chromatography. So that's basically like running it on a gel, but it's doing it a different way with chromatography. But you're sorting things out by mobility on your chromatographic substrate. There will be different substrates, and so you can separate in different ways. But you can do this, so you separate things out, and then once they get to the top or bottom, whatever you want to think of it, of your little column, then you throw them at the detector. You're doing your mass spec. So this is like the two-dimensional analysis. You're sorting things out by, let's say, size, and then you got things of a certain size, and you're throwing them, and you're getting a real precise mass. Or you're sorting them by charge, and then you get a mass, and you're like, okay, it has a charge of +2 and a mass of 219 Daltons. Therefore, it can only be fill in the blank. I have no idea what that would be. All right. So there are a lot of challenges in there. Metabolomics is one of these areas where you can get so much data so fast that it is difficult to analyze potentially. Even if you have a principal component and you can say these 50 lines all appear to be linked, like they grow together, or when these grow, these shrink, and they're connected to each other. Figuring out exactly what each one of those is and how the enzymes that make those could be linked together, that's tricky stuff. Examples of some samples that you could use, fluids, that's very nice. Serum, plasma, urine, for example. Bronchial washes, where they'll sort of put a little bit of soapy water down into a little bit of your lung and try to get that out. See what's going on in there. Do you have like asthma or something like that? Secretions from the prostate, saliva. These could tell you if you have prostate cancer. And yeah, you can use potentially tissue samples directly now. You just have to prepare them first. You have to like isolate the component of the tissue that you want to look at. All right, so there you go. You're going to identify and quantitate, and then you'll make a database of all of the things, like we found all these things. And if you look at assignment A8, that has one of these databases for some proteomic work that we did with sea slug hemolymph, which is like their equivalent of blood. Mollusks are weird, whatever, at least compared to us. And so, yeah, once you got the databases, then comes the informatics part. And it's basically analyzing this, just like how can I best analyze this and make it make sense? Like you got to have a picture of what question am I trying to ask, and then come up with an idea like, okay, like a hypothesis, like, well, if, you know, if this SSRI inhibits serotonin reuptake, then we should see downstream less transmission to serotonin, you know, neurotransmitters, something, receptors, whatever. Less transmission via those, yeah. And so, yeah, that's basically the idea. And then you can plot all these things. You're like, okay, these metabolites go up and these go down when I do this treatment, and then when I do this other treatment, you can plot this. You can actually make something like a family tree from that to show which measurements, metabolites, are going to be most closely related to each other. That kind of gives you an idea of which ones are directly connected. So like if you change this other thing also changes proportionately immediately, you know, stuff like that. Helps you unpack biochemical pathways inside the cell. Usually use only a very small volume, like 0.5 ml. That's fine. You can get 0.5 ml of blood out of a baby. It doesn't really do anything to the baby. You can get it out of a mouse, I think, and it might survive. I don't know. That's probably a little high for a mouse to survive, but still, yeah, pretty powerful. But yeah, if we're talking 100 microliters, yeah, you can definitely get that out of a mouse non-lethally, I believe. So yeah, got a lot of range. Okay. And then based on this, you can go in and look and say, okay, what's different between a cancer cell and a non-cancer cell of the same type? And so you can find differences in all these things, for example. And you can say, all right, a lot of these are just like amino acids or, you know, normal metabolic things like different types of glucose that they might be storing. But there's going to be a relationship because cancers are cells that are growing abnormally compared to the other ones. And so you might expect more things like growth factors, more energy storage, or more energy utilization, and just sort of different metabolism in those, which would help you detect, like from a little blood sample, if there's maybe some cancer somewhere that you should go and investigate. So yeah, powerful technique. All right, so we'll pause right here and then we'll go in for the next section, which I think is the last section. I'll condense it a bit, which is about sort of the data analysis process. So stay tuned.